Alright, this is Jahangir from Medporium, and today we have part 2 of our heart unit, in which we will discuss the basic physiology of the heart. Now, in part 1, we discussed the anatomy of the heart, but we didn't discuss what the heart is made of. Now, the heart is made of cardiac muscle, which is one of the three main muscles that is special to the heart and acts through involuntary movement, meaning that we cannot control this muscle. We can't tell our heart to contract or to relax. Now here's what cardiac muscle, or in this instance, cardiomyocytes may look like. Now as you can see, cardiac muscle is striated, just like skeletal muscle, but the difference between cardiac and skeletal muscle is how they are joined together. Here's a zoomed in picture of what two adjacent cardiomyocytes may look like, connected by an intercalated disc, which is how all cardiomyocytes are connected through. Now, the intercalated disc contains desmosomes and gap junctions. Now, desmosomes are mechanical junctions that bind each cardiomyocyte together and prevents the cardiomyocytes from ripping apart when the heart contracts. Gap junctions are electrical junctions which have channels that allow the passage of ions from the cytoplasm or the inside of the cardiomyocyte to another cardiomyocyte to electrically stimulate any neighboring cardiomyocyte. Just to put that in layman's terms, the gap junctions are just electrical channels that allow the passage of ions from one cardiomyocyte to another, and these ions can include sodium, calcium, and potassium. Now we'll discuss the function of each ion in the electrical conduction of the heart later. Now if we go back to our anatomy of the cardiomyocyte, we can see multiple mitochondria, and this mitochondria is responsible for a process known as aerobic respiration, which is a process of converting glucose to energy or ATP using oxygen. Now compared to skeletal muscle, cardiac muscle contains more mitochondria, which results in the heart not being fatigued at all. Since the heart has more energy generators, it can produce more ATP and never stop beating. The heart also has glycogen, which is a complex carbohydrate that serves as stored energy, and glycogen is just bonded up glucose. Now let's move on to the conduction system of the heart, and that starts with our sinoatrial node, or SA node, which is our internal pacemaker that initiates the heartbeat and determines the heart rate. Now once the SA node generates a signal, that signal from the SA node will spread to the atria, in which the atrioventricular node, or AV node, will fire. Now the AV node prevents any electrical signal from reaching the ventricles by any abnormal route. Now in some cases, if the SA node fails to produce an electrical signal, the AV node can produce it, which results in a heart rate of 20 to 40 beats per minute. To put this in context, a normal adult heart rate is anywhere between 50 to even 100 beats per minute, or 60 to 100 beats per minute. Now after the signal travels through the AV node, it branches out to the bundle of His, which are bundles of fibers that carry an electrical stimulus through the septum. The electrical signal then travels through the Purkinje fibers, which are just nerve-like processes that spread around the ventricles, sending the electrical stimulus in which the heart will contract. This process occurs due to the gap junctions, as we discussed earlier. So in total, the contraction of the heart begins from the SA node, then the AV node, the bundle of His, and the Purkinje fibers. Now let's look at how the autonomic nervous system, or ANS, can control the rhythm and contraction strength of the heart. Now the ANS is broken down into the sympathetic and parasympathetic nervous system. The sympathetic nervous system is our fight or flight response, in which our heart rate increases, digestion is restricted, and in contrast, the parasympathetic nervous system is our rest and digest response, in which the heart rate decreases and digestion is activated. And this is when we're in a relaxed state. Now the ANS connects to the heart through two cardiac centers, the cardioacceleratory center and the cardioinhibitory center. Now by the title of both centers, I'm pretty sure you guessed that the cardioacceleratory is for sympathetic and the cardioinhibitory is for parasympathetic. Now as you can see from the diagram, the sympathetic pathway is shown on the left side and the parasympathetic side is shown on the right side. For the sympathetic pathway, signals from the cardioacceleratory center travel down towards the thoracic portion of the spine, where neurons called preganglion neurons will carry the signal to a sympathetic chain ganglion, where they will synapse with postganglionic neurons, which will carry the signal. Now, hopefully, you didn't get sidetracked by all that medical jargon like preganglion or postganglion, and those are just words just to define the neurons, just to label those neurons. Like pre is before, so before the ganglion, and post after. So you could use medical terminology for big medical words like preganglion or postganglion. Now, once those postganglionic neurons, as well as a bunch of neurons, emerge together, they will form the cardiac plexus, which includes parasympathetic fibers, but for this pathway, our sympathetic postganglionic fibers will move through the plexus and enter the SA node, AV node, atrial and ventricular myocardium, aorta, and other structures. Now, with a sympathetic stimulation, we see an increase in heart rate and contraction strength, as well as a dilated coronary arteries. Now, on the flip side, in the parasympathetic pathway, the preganglionic fibers of the vagus nerve will travel through the cardiac plexus where they will meet the sympathetic fibers and synapse with postganglionic fibers. Now, the postganglionic fibers of the right vagus nerve attach to the SA node, in which the postganglionic fibers of the left vagus nerve attach to the AV node. And as you can see, there's no parasympathetic intervention of the myocardium or ventricles. Now, since we are on the topic of the nervous system and the heart, there are structures known as baroreceptors that are located on the aortic arch and the carotid sinuses, and they detect changes to blood pressure and send that information through the glossopharyngeal nerve and the vagus nerve to the cardiovascular centers in the medulla oblongata, in which the cardiovascular centers will decide a solution and initiate the solution through either the parasympathetic nervous pathway or the sympathetic nervous pathway. Let me break this down for you. If the baroreceptors detect high blood pressure, the glossopharyngeal nerves and the vagus nerves will carry that signal to the brainstem, and the brainstem will figure out a solution and send that signal through the parasympathetic pathway to decrease the heart rate so that in turn blood pressure can decrease. Now, it'll also send a signal to the arteries for dilation, since this will also lower blood pressure. Now, we must be aware that the heart can contract on its own even without the nervous system. The nervous system can only make the heart beat faster or slower, but it cannot produce a heartbeat. This is the reason why the heart can continue to pump even out of the body. To put this in context, let's look at Bob. Now, Bob is at school and he has a normal heart rate of 78 beats per minute until he realizes that there's a major grade essay due in 2 hours and he hasn't even started it. Bob then experienced a tremendous amount of stress, and this activates his sympathetic nerves, more specifically the cardioacceleratory center, which increases his heart rate and blood pressure. And obviously, since he's stressed out, how is he going to complete this major grade essay in 2 hours, which might change his whole grade? Now, his baroreceptors in his heart and cardiac sinuses detect this rise in heart rate and blood pressure and send a signal from the glossopharyngeal and vagus nerve in an attempt to bring the heart rate and blood pressure back to normal. But the cardiovascular center in the medulla ignore this request because the quote-unquote threat is not over. Because the brain interprets Bob's stress on this essay as a threat to Bob, which is why the medulla will continue with a sympathetic response so that blood can be pumped to the skeletal muscle and restrict to his digestive system, since it is more important for his legs to be oxygenated than his digestive system in events such as running from a dangerous animal or focusing on his essay. Now, Bob finally finishes his essay and turns it in and receives a 95, in which he feels relieved and relaxed. And when this occurs, the cardiovascular centers acknowledge the request from the baroreceptors to lower the blood pressure and heart rate, which results in a stimulus sent from the vagus nerve to the SA node, which also results in a decrease in heart rate. So basically, the cardiovascular centers say, okay, yeah, you're right. We're in a relaxed state. We don't have any threat anymore. The essay is already over, so we can just lower the blood pressure and go back to normal. So to summarize, the vagus nerve slows our heart rate down if we're relaxed, and the postganglionic nerves that are sympathetic speeds up our heart rate if we're not relaxed, such as exercising or if we're in stress. Now, the baroreceptors detect any abnormality in the blood pressure and heart rate and tries to bring the body back to normal, which is a process known as homeostasis. Now, the heart can beat on its own, but it will beat at 100 beats per minute. The vagus nerve slows this number down by a normal 50 to 90 beats per minute, assuming that you are an adult. Now let's discuss the pacemaker action potential. But before we do that, I want to discuss what the membrane potential is. Now, membrane potential is the difference in electrical charge from the outside and inside of the cell. The membrane potential is measured by millivolts, and this membrane potential depends on the ions entering or exiting the cell membrane. For example, as sodium and calcium flow into the cell, the membrane potential becomes more positive, and obviously so because sodium and calcium are both positive ions. And if the potassium flows out of the cell, the membrane potential will be more negative. Now, one thing to note is that the SA node, cardiomyocytes, and neurons all have different electrical behaviors and action potentials. It is very important that you do not base your knowledge on the pacemaker action potential with the action potential of the cardiocytes or neurons and vice versa. Now, before we discuss the action potential, let's discuss systole and diastole, as well as depolarization and repolarization. Now, systole is defined as a phase of contraction of the heart, which results in the flow of blood through the chambers of the heart, as well as the aorta. Now, in systole, the semilunar valves, which are basically the aortic and pulmonary valves, open, and the atrioventricular valves, which are the tricuspid and the mitral/bicuspid valve, close. Now, diastole is a phase of relaxation in which the heart is not in contraction and the atria and ventricles fill with blood. Now, in this phase, the atrioventricular valves are open and the semilunar valves are closed. Now, just remember, when I refer to atrioventricular valves, I'm talking about the tricuspid valve and the mitral valve. And if I'm talking about the semilunar valves, I'm talking about the aortic and pulmonary valves. Now, systole is initiated through a process known as depolarization, and this starts when the SA node is stimulated, which initiates a muscle contraction. And diastole is initiated through repolarization, which is when the SA node recalibrates and has no muscle contraction. Now we can move on to our discussion of the pacemaker action potential, and here is a picture of what the pacemaker action potential may look like. Now, to start, we need to look at the threshold potential, and this is the millivolts the membrane potential must be in order for the SA node cells to be stimulated. The threshold potential for our pacemaker cells is about negative 40 millivolts, and the negative 60 millivolts is known as our resting membrane potential. And as the name implies, this is the membrane potential in millivolts that our cell is at when the cell is resting. Now, to meet the threshold of negative 40 millivolts, the sodium channels in the pacemaker cell membrane open, allowing for the slow inflow of sodium, and when the membrane potential actually reaches negative 40 millivolts, the calcium channels open, allowing for calcium to flow into the cell, which results in this rapid depolarization. And this is because calcium has a positive charge. It has a positive 2 charge, which will make the membrane potential more positive. Now, once we reach the peak membrane potential, the calcium channels close and the potassium channels open. But in this instance, potassium flows out of the cell, resulting in repolarization. And this makes sense because potassium is positive. And if we have an ion that's positive and it's leaving our cell, we obviously become more negative until we reach our resting membrane potential. Now let's discuss the cardiac rhythm, and to do that, we need to look at this diagram showing the action potential of cardiomyocytes. Let's look at phase 0, in which our voltage-gated sodium channels open, and sodium will flow through the channels since they are open. Now, the reason why you see a line exponentially growing is because the sodium has a positive charge, and as we can recall, the resting membrane potential is negative, which means that when sodium enters the cardiomyocyte, it makes the membrane potential less negative and more positive, which is what depolarization is. Now, when sodium is entering the neurons, this results in depolarization. And in phase 1, the sodium channels will actually close and the membrane potential will peak at a positive 30 millivolts. The next phase is phase 2, in which potassium channels and calcium channels open in the cell membrane. Now, in phase 2, potassium leaves the cell, and normally when potassium leaves the cell membrane potential becomes more negative because potassium being positive is leaving the cell. But since we have calcium entering the cell, which is positive, this almost counterbalances or cancels that amount of potassium leaving the cell, so this results in what's known as a depolarization plateau. Now, this depolarization plateau is seen in cardiac muscle cells rather than skeletal muscle cells. The next phase is phase 3, in which we see a rapid repolarization of the cardiac muscle cell due to the calcium channels closing, which results in a more negative membrane potential. Now, the final phase is phase 4, in which cardiac muscle cells return to resting membrane potential of about negative 90 millivolts. Now let's discuss the electrocardiogram, or ECG or EKG. Now, electrocardiogram is a diagnostic test that detects electrical currents generated by the heart through electrodes attached to the wrists, ankles, and chest to record the same electrical activity of the heart. Now, there are different types of EKGs, such as the exercise EKG or the stress test, the Holter monitor, and even the EKG on your Apple Watch. But the EKG we will focus on is known as a 12-lead EKG, which is one of the most common EKGs used for the diagnosis of potential cardiovascular diseases. Now, here's a diagram of what the EKG may look like. Let's read this from the left to right, just like a book. The first structure we see is known as a P wave. Now, the P wave is generated when the SA node signal spreads through the atria, which is filled with blood, resulting in the depolarization of the atria. Now, atrial systole starts about 100 milliseconds after the P wave, in which blood will rush into the ventricles and the atrioventricular valves open. The next structure we see is known as the QRS complex, in which the AV node fires, resulting in ventricular depolarization. Now, the Q wave occurs when the action potential reaches the interventricular septum, causing the depolarization of the septum. The R wave occurs when most of the ventricle has been depolarized, and the S wave concludes the depolarization of the ventricles. The final wave is the T wave, in which ventricular repolarization occurs and diastole follows. Now, one thing to note is that the depolarization of the atria and ventricles occurs in a base to apex motion, or top to bottom, but the repolarization of the ventricles occurs in the apex to base motion, or bottom to top. Now, there are other parts of the EKGs that physicians must interpret besides just the P wave, the T wave, and the QRS complex. Now, first, there's the PR interval, which demonstrates the conduction of the AV node going from atrial depolarization to ventricular depolarization, with a normal range being 120 milliseconds to 200 milliseconds. We then have the QRS interval, in which the interval of the ventricular depolarization and the normal range is 80 to 100 milliseconds. Finally, we have the QT interval, which is the interval going from ventricular depolarization to repolarization, and the normal range for adult men is 350 to 450 milliseconds, and for an adult woman, it's 360 to 460 milliseconds. We also have segments on the EKG, which are just flat lines, as you can see, and these include the PR segment and the ST segment. Now, to put everything into context, let's discuss the phases of cardiac cycle. Now, the first phase is known as ventricular filling, and as the name suggests, this is when the ventricles fill with blood. Now, ventricular filling is broken down in 3 components. The first component is known as rapid filling, and this is, as the name implies, when blood rapidly fills the ventricles due to high pressure of the atria that has been built up, which causes the AV valves to open. The second component, which is also known as the middle of diastole, is called diastasis, and this is when the filling of the ventricles has slowed, but before the atria has contracted. The last component, which is also known as atrial systole, is when the atria contracts, and this results in most of the blood from the atria moving into the ventricles. Now, the second phase of the cardiac cycle is known as isovolumetric contraction. And in this phase, the pressure of the right and left ventricles increases due to the AV valves being closed, and pressure builds up in the ventricles until the ventricular pressure is higher than the atrial pressure, in which we move on to our third phase, which is known as ventricular ejection. Now, this is when the ventricles contract and the pulmonary and aortic valves open, which results in blood flowing out of the heart. Now, once this occurs, we move on to our final phase, which is known as isovolumetric relaxation. And this is when all the valves are closed and blood flows into the right and left atrium from the inferior and superior vena cava and pulmonary veins, respectively. Now, after isovolumetric relaxation, we move back into ventricular filling, and this occurs due to the pressure built up in the atria that causes the blood to move to the ventricles. Now, you must understand that blood, as well as everything else, moves from a high pressure environment to a low pressure environment, or a high concentration environment to a low concentration environment. And this high pressure to low pressure is known as a pressure gradient, and is established when you breathe in and out, for example. Now, when you inhale, you are creating a pressure gradient by expanding your lungs, which increases the volume of the lungs. And if you remember Boyle's law from chemistry, higher volume equals lower pressure, which results in the lungs having lower pressure since the lungs are expanding and volume is increasing and the air around you has a high pressure. And this is how air enters your lungs. Now, air will not be able to enter your lungs if you just open your mouth, because the pressure of the environment and your lungs are the same. So that's something you really need to understand. Now, at this point, after our discussion of the phases of the cardiac cycle, you may be wondering why diastole occurs after the T wave and during the P and Q wave instead of just the P wave, because it just sounds a lot easier, or why systole occurs only in the R, S, and T waves instead of just the QRS complex. Well, this is because it takes time for the signal produced from the SA node to travel through the AV node, bundles of His, and Purkinje fibers. This delay of a mere tenth of a second accounts for why 4 phases occur during different parts of electrical rhythm. Now let's discuss the different sounds the heart makes, and I'll have the cardiac cycle diagram as a reference. Now, the main heart sounds include S1 and S2, which create that familiar lub-dub sound. Now, the space between S1 and S2 is systole, as you can see from the diagram, and the space between S2 and S1 is diastole. Now, the S1 sound is produced by the closure of the AV valves, as well as blood rebounding to the AV valves, which will occur during isovolumetric contraction, in which the AV valves will be shut, and blood will push against the valves, resulting in this S1 sound. Now, the S2 sound is produced when the semilunar valves close and blood rebounds to the semilunar valves during the start of diastole, which is our isovolumetric relaxation. Now let's move on to S3 heart sound, also known as ventricular gallop, which is during diastole and after the S2 heart sound. This heart sound occurs during rapid filling and diastasis of the ventricular filling phase, in which this large influx of blood from the atria to the ventricles produces this vibrating sound that can be heard through the sound of Kentucky. Now, the final heart sound is called S4, which is also known as atrial gallop, and this occurs before S1 during the atrial systole of ventricular filling, in which the atria creates a forceful contraction due to a non-compliant or stiff ventricle. Now, one thing to note is that the S3 heart sound is totally normal in children, healthy young adults, pregnancy, and athletes. And the S3 heart sound also can be an abnormality or a symptom of severe mitral or tricuspid valve regurgitation, cardiomyopathy, and congestive heart failure if heard in elderly patients. Now, the S4 heart sound is usually an indication of pathological disease, which includes diastolic heart failure, left ventricular cardiomyopathy, or hypertrophic cardiomyopathy, hypertension, or aortic stenosis. So to recap, S1 and S2 heart sounds are the universal heart sounds you will encounter, and it is identified by a lub-dub sound, and those are the perfect, healthy, and normal heart sounds. Now, S3 heart sounds can be normal and pathological at the same time, whereas the S4 heart sound will always be an indication of an abnormality or some sort of pathological disease. Alright, and unfortunately, that concludes our heart physiology video. I hope you learned something today, and as always, a Pomodoro a day will make you a doctor someday. I'll see you next time.